Pues los organizadores de esto se han quitado el marrón y de la presentación y me lo han cascado a mí. Pero bueno, pues nada, hoy vamos a ver el proyecto de tesis de Martín López, que empezó la tesis en enero, ¿no? Con una FPU y ahora nos va a contar de qué va.
Irina Martín López, and my PhD is about the ecology of West Nile virus in Andalusia, strategies to reduce the risk of transmission to humans. And my supervisors are and Martínez de la Punta. First, I would like to talk a little bit about me. Thank you. Like mammals, birds, reptiles, or amphibians. In my case, I'm going to study the interactions between West Nile virus and avian malaria parasites transmitted, in both cases, by mosquitoes to different species of birds, and in the case of West Nile virus, also transmitted to humans and horse. West Nile virus is a zoonotic flavivirus transmitted mainly between birds and mosquitoes. But mosquitoes can also transmit the virus to horse and humans. But these hosts are considered dead-end hosts because they cannot transmit again the virus to mosquitoes. That is what we consider the humans and horse dead-end hosts of the virus. The West Nile virus was first discovered infecting humans in Uganda, and we detect the virus in the 1962, first circulating in Europe in birds, humans, and also horse. But the virus started to gain importance in the 1999, where its first introduction, and only in few years, the virus spanned across all the American continent, from New York to California. The virus produced more than 2,300 deaths, human deaths, and around $800 million on hospitalized West Nile virus patients. In addition, the virus produced high ecological costs because produced the decline of different bird populations, such is the case of the Corvus brachyrhynchus or American crow, that the virus produced the decline by up to 45% of the populations. Here in the graphic, we can see different localities of the Corvus brachyrhynchus, and after the introduction, that is the red line, in the graphics of the West Nile virus, all the populations decline. We can see also in the histograms the West Nile virus human case. So these population declines occur at the same time that we have West Nile virus human case. In Spain, the West Nile virus was first detected circulate in the 2003 in birds and in 2004 in humans. In Andalusia, we detect the West Nile virus in humans and horse in the 2010. And since then, the virus has been circulating with variations in the West Nile virus prevalence in different years and between also geographical regions. Et al. Have found variations in the seroprevalence of West Nile virus between different years. In the case of horse, since the detections in the 2010, there are also variants in the West Nile virus seroprevalence. But in the case of humans, we only had 6 human cases until the 2020, where we have a human large outbreak with 77 infections and 8 human deaths. Since then, the virus has been circulated between birds, horse, and humans in 2021, 2022, and 2023, But in the 2024, we have the largest human outbreak here in Spain with more than 150 infections and around 20 deaths, human deaths. This is the current situation of the West Nile virus in Europe and is worsening, because this data reveals that the surveillance of the virus is not enough for decrease the West Nile virus human case. So we need a better understanding of how the ecology and evolution of this virus are occurring for decreasing these West Nile virus human outbreaks. Here in Spain, the Culex mosquitoes can also transmit other pathogens, such as the case of the avian malaria parasites of the genus Plasmodium. Malaria parasites exhibit a wide ecological diversity, while some of these parasites can infect mammals, such as the case of the human malaria. Other malaria parasites can infect birds. For example, the human malaria life cycle is similar to the best malaria life cycle, where we have the mosquitoes that are the definite host of the parasite because the sexual reproduction of the parasites occurs in them, and are the vector of transmission pathogen to the birds that are the intermediate hosts. Due to their similarities in the transmission of West Nile virus and avian malaria, because we can see that they have similar transmission cycles and they share vector species, in our case Culex pipiens and Culex perexivus. Both pathogens are transmitted by these two vector species to birds, and in the case of West Nile virus, also to horse and humans. So due to their similarities, studying together West Nile virus and avian malaria provides a deeper understanding of the ecology of this vector-borne disease. And in my case, studying avian malaria parasites provides a better understanding of the transmission dynamics of West Nile virus. So the advantages of using Plasmodium are that we have a huge diversity of Plasmodium lineages, and we can work with how different factors affect the prevalence, but also the richness and diversity of these parasites in the case of Plasmodium. But in the case of West Nile virus, we can only work with West Nile virus prevalence in the case of mosquitoes, because we have different lineages of the virus, but here in Spain, in the southwest of Spain, we only have one lineage circulating, so we can only work with West Nile virus prevalence in mosquitoes. In the case of birds, in seroprevalence. In addition, Plasmodium show higher prevalence in mosquitoes than West Nile virus, so we have a stronger detection signal of the parasites instead of the West Nile virus, and also the Plasmodium produce chronic infections in birds, while the West Nile virus produce short viremia. That is the reason that we have to detect the antibodies and not the real infection. So that is the reason that I say seroprevalence instead of prevalence. Furthermore, detecting Plasmodium in birds is cheaper, so because we need a conventional PCR, and in the case of West Nile virus, we need high biosafety labs for detecting these antibodies in the case of birds. So Plasmodium could help us infer West Nile virus patterns due to their similarities in the transmission cycles. And for example, if we detect areas with high circulation of Plasmodium and these areas are the same of the high circulation of West Nile virus, we can infer West Nile virus patterns and also predict West Nile virus outbreaks. And this could be helpful not only in our study area, but others in other areas that they have a lot of data of Plasmodium, but not from West Nile. In most cases, this spatiotemporal variation in the prevalence can be explained by different factors. These interactions between birds, mosquitoes, and Plasmodium and West Nile virus occur in an environmental context. That is, the intrinsic factors like abiotic factors. Previous studies have analyzed the interactions between different environmental factors and how these environmental factors affect the Plasmodium prevalence, richness, or diversity in birds. For instance, some studies have found a positive relationship of temperature and a negative relationship of precipitation in Plasmodium prevalence, richness, and diversity in birds. But these depend on the elevation, producing altitudinal variations on Plasmodium communities. Other studies analyzing the land use have found that rural environments present lower Plasmodium prevalence and lower Plasmodium richness than natural and rural environments, producing spatial variations also on Plasmodium communities. In the case of West Nile virus, there are a lot of studies analyzing how these environmental factors affect the West Nile virus seroprevalence inverse. In Spain, some studies have found that temperature is positively related with West Nile virus seroprevalence inverse and also the distance to water source. But despite the importance of mosquitoes in the West Nile virus and avian malaria transmission, there are few studies analyzing how environmental factors affect disinfection patterns in mosquitoes. And all the studies doing in birds, like how environmental factors affect disinfection patterns in birds, conclude that these environmental factors are shaping the mosquito abundance. So we need to better understand of how these environmental factors affect the infection patterns in mosquito vectors. Few studies, like Ferragut et al., have found that the distance to water source seems to be important for understanding the plasmodium infection patterns in mosquitoes. But to the best of our knowledge, no one has studied yet the relationship between other environmental factors like temperature, land surface temperature, or vapor pressure with plasmodium infection patterns in mosquitoes, despite the importance of these environmental factors in mosquito abundance and also in mosquito activity patterns. In the case of West Nile virus, only few studies analyze how these environmental factors affect the West Nile virus prevalence in mosquitoes, at least in Europe. So we need a better understand. The second factors are the biotic factors, because the interactions between birds, mosquitoes, and these two pathogens occur in a context of bird communities and vector communities, where the pathogens can interact with different bird species and different mosquito species. Previous studies analyzing how pathogen prevalence is related with bird richness have found different results. The first one have found that the bird richness is negatively related with the pathogen prevalence. This is because lower fraction of competent hosts in the community produce lower transmission success and therefore lower Plasmodium or West Nile virus prevalence. For instance, Suat and Carlos have found a negative relationship between the bird richness of West Nile virus in humans in the United States. But other studies have found the opposite result. That means a positive relationship between the bird richness and the pathogen prevalence. For instance, Ferragut et al. Have found that richer avian communities have a higher West Nile virus seroprevalence inverse. So we need a better understanding of how this pathogen prevalence is related with the bird richness. The third factors are the intrinsic factors, because pathogens are going to interact different between different species and also between different individuals from the same species. In the case of birds, for example, pathogen infections depends on three different variables: the host susceptibility, the attractiveness to mosquito host, and the host exposure to mosquitoes. The host susceptibility varies between individuals and also between species, and is essential for understanding amplification dynamics and improving surveillance strategies. In the case of the attractiveness to mosquito, the mosquito have different sensors to detect their host, and their host can produce different cues for mosquitoes, that is CO2 or body odors. In the case of birds, the body odor mainly produced by the uropial gland secretions. These uropial gland secretions can be modified by the interactions with other organisms, and such is the case of the interactions of pathogens and these uropial gland secretions. Pathogens such as avian malaria can modify the chemical composition of these uropial gland secretions to attract more mosquitoes and therefore to be more successful in their transmission. For instance, Diaz Fernandez et al. Have found that birds infected with Plasmodium were more attractive to mosquitoes than birds uninfected with Plasmodium. But other studies have found the opposite results or no significant results at all. So we need also a better understanding of how pathogens are related to these uropial gland secretions. The other organisms that can modify these uropial gland secretions are the microbiota of the. We have different microbiota depending on the individuals and on the species, and there are some studies analyzing how uropial gland microbiota or skin microbiota modify this bird odor, but they are still lacking a knowledge of how microbiota from other parts, like cloacal microbiota, can modify this bird odor. The third variable is the host exposure that depends on the life history trace of the birds that can modify the host exposure to the mosquitoes vectors. These life history traits can be separated in physical attributes of birds, such as body mass. For instance, Figueroa et al. Have found that larger species present more West Nile virus seroprevalence, and the other types are the ecological traits. Such is the case of the migratory behaviors of birds, where some studies have found that migratory species present more West Nile virus seroprevalence than other species. Therefore, we have a lot of factors interacting in a complex network at the same time, and these are producing the spatiotemporal variation in the West Nile virus seroprevalence or prevalence and Plasmodium prevalence. In the case of West Nile virus, if different factors align at the same time, we are going to have high risk for transmission this West Nile virus to humans. For instance, if we have increased viral circulation and greater mosquito abundance and other factors, we are going to have a human outbreak. So the objectives of my thesis is to identify these factors that are affecting the transmission dynamics of West Nile virus and producing these ecological chains that favor the spillover of West Nile virus to human population, causing these epidemic outbreaks. And for that, we are using also the Plasmodium parasites for better understand how this West Nile virus transmission dynamics is occurring. For that, we pretend to do 6 different chapters divided in 2 sections. In the first section, we are going to focus in the interactions between West Nile virus and mosquito vectors. In my first chapter, we are going to study the interactions between avian malaria parasites and mosquito vectors in an environmental context. In the second chapter, the interactions between West Nile virus and mosquitoes, but also the interactions with birds communities, mosquito communities, and environment. And in my third chapter, we are going to study the interaction of these 2 pathogens in mosquito vectors. In the second section, we want to focus in the interactions of these 2 pathogens and vertebrate hosts, in this case, the birds. In my fourth chapter, we are going to study the interaction of these 2 pathogens in birds, but also the interaction of a bird community, vectors communities, and environment. In my fifth chapter, the interaction of avian malaria parasites and birds. And in my last chapter, how West Nile virus interact with different bird species. For that, we are going to focus my thesis in the southwest Andalusia due to the high West Nile virus risk that this area have. And also, mostly of my thesis is going to focus in the periods of the 2020 to the 2024 due to we have 2 years with large human outbreaks, the 2020 and the 2024, and 3 years with lower West Nile virus incidence. So we have 2 different years to compare, ones with higher West Nile virus incidence and others with lower West Nile virus incidence in humans. For that, we sample different birds depending on the species with different methods, and in the case of mosquito, with bee traps, and then we separate the mosquitoes based on the morphology in Culex pipiens and Culex plexigus. For detecting West Nile virus in the laboratory, in the case of mosquitoes, we use a real-time PCR, but in the case of birds, we have to detect this due to the short viremia the virus produced in birds. And in the case of Plasmodium, we use unconventional PCR, and then we do the sequencing for detect the lineage of Plasmodium that are infected that sample. Then to talk a little bit about my 6 chapters. In my first chapters, as I say, environmental can shape Plasmodium communities in birds, but little known is about how these environmental factors affect the Plasmodium communities in mosquitoes. So we want to which environmental factors are predicting this prevalence, richness, and diversity in Plasmodium in 2 mosquito species, Culex pipiens and Culex plexigus, and also to compare the Plasmodium lineage communities between the two vector species. This chapter is currently under review in scientific reports. For that, we sampled around 100,000 mosquitoes of Culex pipiens and Culex prexiguus in Huelva and Seville in 2021 and 2022. And we found that Plasmodium prevalence was higher in Culex pipiens and Plasmodium richness was higher in Culex prexiguus than Culex pipiens. In addition, we found that environmental variables were significantly related with different Plasmodium communities variables. For instance, we found that Plasmodium prevalence in Culex pipiens was positively related to pH, and Plasmodium prevalence in Culex prexiguus was positively related with temperature. These two environmental variables are positively related with mosquito abundance, so that could be the reason that we found these significant interactions. In addition, we found that geographical distance was significantly related with Plasmodium communities in Culex pipiens, but not in Culex prexiguus. That means that Culex pipiens populations that are near present more similar Plasmodium communities than those that are further apart, but not in Culex prexiguus. This could be because Culex pipiens and Culex prexiguus present different population structure. Culex pipiens is more wild distribution, while Culex prexiguus have more patchy distribution in the map. And this could be because they present different habitat preference. For example, Culex pipiens is more generalist, while Culex prexiguus prefers habitats near rice fields. So this difference in habitat preference between vectors could be the reason that we found different population structure and therefore different Plasmodium communities depending on the vector species that we are studying. In my second chapter, there also lacking of knowledge of how these biotic and abiotic factors affect the the West Nile virus prevalence in mosquitoes. So in this chapter, we want to assess the influence of different environmental variables and also biotic factors, diverse communities and mosquito communities, on West Nile virus circulation under different levels of West Nile virus human incidence. So for that, we are going to use data from Culex prexiguus for 3 years with lower West Nile virus incidence and 1 year with a large human outbreak. Preliminary results show a positive relationship of the maximum temperature and West Nile virus infection rate in mosquitoes and a negative relationship with mosquito richness. In my third chapter, as I say, working with Plasmodium might help us infer West Nile virus patterns and could potentially contribute to predict future outbreaks in humans. So in this chapter, we aim to assess if there are a significant correlation between Plasmodium prevalence and West Nile virus prevalence by year, and also to evaluate the predicted availability of Plasmodium to detect the human West Nile virus outbreaks. So for that, we are going to use Culex prexiguus, that is the main vector of the West Nile virus, in Huelva and Seville between 2020 to 2024, 2 years with large human outbreak and 3 years with lower West Nile virus incidence in humans. Exploratory analysis revealed a positive relationship between Plasmodium prevalence and West Nile virus prevalence, but only in the year with large human outbreak, in the 2020, but not in 2021 and 2022. In my, in my first chapter on my second section, we also know that working with Plasmodium and West Nile virus can reveal West Nile virus patterns, but in this case, inverse. And also, studying together the West Nile virus, avian malaria, and also biotic and abiotic factors could be more informative than study alone how environmental factors modify the Plasmodium infection inverse or how biotic factors modify these infection patterns inverse. So we want to understand which environmental and biotic factors together predict the Plasmodium prevalence, richness, and diversity inverse, and also the West Nile virus seroprevalence together, and also to assess the spatial and temporal variation between West Nile virus and Plasmodium. So this chapter is similar to the 1, 2, and 3 in mosquitoes, but this all together in bears. And for that, we are going to use samples from house sparrows, sample between 2020 and 2024 again, in Huelva and Seville. But we are now working in the laboratory with these samples. In my 5th chapter, as I say, there are different results on how parasites can modify the chemical compositions of the European gland secretions. And also, little note is about of how urbanization and cloacal microbiome diversity can modify this body odor. So we want to identify the role of blood parasite infections, habitat, and cloacal microbiome diversity on the chemical composition of the European gland secretions. So for that, we use house sparrows again, sampled in Huelva and Seville in 6 localities in 2023. And we use blood for detecting Plasmodium, the cloacal microbiome diversity, and also we analyze the chemical composition of the European gland secretions in a chromatogram. Preliminary results show that urban environments have lower chemical compound richness in the European gland secretions than natural and rural environments. And also, we found that Plasmodium-infected bears present more chemical compound diversity in the European gland secretions than uninfected bears. Finally, the ecological and evolutionary factors driving this spatiotemporal variation in West Nile virus incidence remain unclear because there are a lot of factors interacting together. So we want to examine the roles of phylogeny, life history trace, and habitat in use and temporal variation in West Nile virus exposure. This chapter is also currently under review, and for that we use a long-term data between 2003 and 2024, and we sampled around 20,000 individuals belonging to 48 species. We found a variation in the West Nile virus seroprevalence across different years, and the year 2024 were the year with the most West Nile virus seroprevalence. That is the year with the largest human outbreak here in Spain. In addition, 41 of 48 species present West Nile virus seroprevalence antibodies, and Gruiformes, Charadriformes, and Passeriformes were the orders containing the species with the highest seroprevalence. In fact, Bayesian models reveal a strong phylogenetic effect on the West Nile virus seroprevalence. These Bayesian models shows different interactions between life history trace of the bears and West Nile virus seroprevalence. For instance, body mass was positively related with West Nile virus seroprevalence, and this could be because larger species produce more CO2, more heat, or more body odor signals that are detected of the mosquitoes, and this could be that we found this positive relationship. Other example is that we found that short-distance migrants present more West Nile virus seroprevalence than sedentary bear species, and this could be migratory bears have higher exposure to this virus due to being in different environments. Finally, we found a significant interactions between bear habitat and years without or with human outbreak. In fact, we found that bears from urban habitats present more West Nile virus seroprevalence in years with human outbreak than years without human outbreak. But bears from natural habitats, we didn't find significant difference between years without human outbreaks and years without human outbreak in the West Nile virus seroprevalence. These results reveal the importance of urban bears epidemiology and also suggest that the human outbreaks are occurring due to an increase in the viral circulation, but only in urban areas and not a general increase in the viral circulation in the geographic region. Finally, I would like to talk a little bit about my chronogram. I pretend to finish my chapter 1 and 6 the next year because these chapters are currently under review. The chapter 2, 3, and 5 in the third year, and my chapter 4 in the final year because we need to do the laboratory analysis and statistical analysis for that chapters. I pretend also to do a research state with Dr. Nicola Clark in the University of Queensland, and we pretend to do population dynamics models in mosquitoes and how West Nile virus can affect these mosquito populations. As you have seen, much of my data used in my thesis span many years, and this couldn't be possible without the collaborations of many people from Estación Biológica de Doñana, Ciberspep, Diputación Provincial de Huelva, Instituto de Salud Carlos III, Universidad de Granada, and INEA. So, thanks to all my collaborators, and thanks to all of you for your attention, and I will be happy to answer all your questions.
Thank you, Anna. Very clear. Do we have questions for Anna?
So, very interesting and nice talk, beautiful slides.
Thank you.
So do you think that the prevalence of the two diseases is just correlated, or if there's an interaction between them, like the health effects of one facilitate or inhibit the other?
You mean that if we know that Plasmodium can interact with West Nile virus, reducing or amplifying the prevalence? We really don't know if, I don't know if Plasmodium is interact with West Nile virus. I think they have not interact big cells of the birds, and West Nile virus is more in the brain and other tissues. So I think that they are not interacting, but
the immune system would
yes, probably
be reacting independently.
I'm going to read about that, but it's interesting. Could be, but I don't really know.
Wondering, because now there's, so I don't know that much about this, but you hear in the news about the bird flu outbreak, and I was wondering if that would have any impact on your system.
I don't really know. Jordi says no.
Oh, sorry. I'll pass the microphone.
They are completely different parts of the immune system that are involved. One is intracellular parasite, so there are different components. There is no interaction at immune level. So the hypothesis is that since ecology is very similar, the patterns of transmission can be very similar. That's the hypothesis that Anna should test in the PhD. So the idea is if it is possible to infer what can happen with West Nile virus before it arrives using Plasmodium in the areas where the virus has not arrived yet.
Hi, Anna. Great talk. So you went a little quickly. You have many chapters and hypotheses or aims of the chapters, so I got a little bit confused. So there's so you have a massive sampling of over almost 200,000 mosquitoes, and you've run some models trying to test the environmental variables that could explain seroprevalence.
Yes,
and
in the
you've
first
shown, if I remember correctly, some results with NDVI and temperature.
Yes.
And I was wondering, how you built those models, how many variables you considered, how you come up with these results? Because it's a very nice result, but how do you get to that?
We made linear or generalized mixed models for the prevalence, richness, and diversity separately in Culex pipiens and Culex perexius. So we make 6 different models, and we select the best model with the IC criterion, and we use, these environmental variables for the models. Okay. So we select the best model. How is the best model? Explaining the Plasmodium prevalence or Plasmodium richness or Plasmodium diversity in Culex pipiens and also in Culex perexius.
Okay, thanks.
Well, okay, if there are no more questions, please thanks Anna again.